Hi, everyone. Before I start, I just want to say thank you to Joanna and Claire who helped to reschedule my talk because I left my laptop on airplane and luckily got it back on Tuesday from Lost and Found. I really appreciate your help. And okay, so I changed my title a bit shorter, but the idea is similar. We want to try to identify the molecular evidence left by polygenic adaptation. I'm a PhD student from Doug Edge's lab at USC. And so we know that phenotypic adaptation could happen across different evolutionary scales. And at microevolutionary scale, it usually happens over a relatively short time period. Like think about the human skin pigmentation variance depending on where our ancestors lived. And usually people, this type of difference could be traced back to allele level by comparing the genome of individuals within the same species. And at the macroevolutionary scale, it usually could happen over a longer time scale. Think about the mammal, the body size between mammals like an mouse and an elephant. And usually people try to search for molecular evidence on the genome by comparing the genome from individuals of different species. And now that we know which type of phenotypic adaptation we are looking for, the next question is to figure out how to detect the signal on the genome. And in the field of molecular evolution, there are three kinds of classic methods. They're polymorphism-based, divergence-based, or methods that combines both. So Tajima-STI is an example of polymorphism-based. Basically, the idea is to compare the total number of segregating sites and the average pairwise sequence between the genome within the species. And DN-DS ratio compares the rate of non-synonymous mutation to synonymous mutation to identify a selection. And McDonald-Friedman test combines both the information from polymorphic site and divergent site and to see if there's a selection signal. We'll come back to MK test in a bit and dig into how it works in detail. And for now, let's just zoom in to an icon. Oh, sorry.
The top bar is blocking the title.
Oh, sorry. I should move the mouse.
No.
Cool, thank you. And it is an iconic example in biology, the comparison between human and chimp. So people have found that the genomes between human and chimp are about 99% identical. And that raises an interesting question. Since our genetic data are so similar, then how could we end up looking and behaving so differently? For the past few decades, people have tried to search for molecular evidence to explain this divergence. And here I just listed 3 examples to try to explain it. But until today, there's no single mechanisms that can fully explain the divergence between human and chimpanzee. That's probably because that many traits are polygenic, which means that a trait could be controlled by hundreds or thousands of genes, and each gene just contributes a small effect. So on the right side is an example of GWAS association for height in Europeans. Each dot here is an associated allele with their frequency in the population on the x-axis and their corresponding effect size on the y-axis. And you can see how many associations here, and most of them just have small effects. This is the typical pattern for polygenic trait, which makes detection signal really challenging. And while polygenic traits are highly complex, which also means they are highly tunable by evolution. And many polygenic traits are shaped by stabilizing selection. So under a stabilizing selection, the individuals with extreme trait values tend to be removed. And those individuals with average trait values, they tend to have higher reproductive success. So if you're experiencing stabilizing selection, we could use a normal distribution to model the mapping relationship between phenotype and fitness, and the fitness would just be the probability density of the corresponding phenotype. In this project, the question we try to answer is that if we have a polygenic adaptation experiencing under stabilizing selection, could we detect between species, could we detect any molecular signature on that? And we started with the population genetic model. So we simulate a structured chromosome with a functional part or axons or genes, whatever you call it. So the dark navy part are the functional parts, and the rest parts are just neutral parts. And we initialize this chromosome as a wild type, which means without any mutations on it. And as the evolutionary process goes on, the mutations just show up stochastically across the chromosome. And here we defined two types of mutations. M1 mutation is a neutral mutation. It has no effect on the trait value, and it can occur anywhere on the chromosome. And M2 mutations are the trait-associated mutations. So it can only happen at the functional area. And each time when an M2 mutation occurs, we draw an effector size from the normal distribution with variance as sigma squared. And to make our simulation more accurately reflect the mutational process in the coding sequence, we also model the position, the codon position with different probability of M2. So sorry, for the first and second positions in the codon, we let the probability of M2 mutations as 1/2. That is because in real life, most of the mutations that happen at these two positions will lead to a change in amino acid, or you can call it non-synonymous distribution. And those mutations happen at the third position, usually won't lead to any amino acid change, and the probability is reduced to 1/6. And our evolutionary process basically mirror the phylogenetic relationship between human and chimp. And we started with the single ancestral population. And at the time point of speciation, we introduced a new population and let them just evolve divergently. At each generation, we get the information of genotype, which is basically the effector size of M2 mutation. And we get the sum of it, take it as a breeding value or phenotype, because here we don't consider any environmental factor. And the fitness would be the corresponding density of the phenotype. The three highlighted parameters here are the key parameters in our simulation. So VS here represents the selection strength. Imagine that under a stabilizing selection, smaller VS will make the distribution narrow. So that indicates a strong selection. And sigma square is the variance of the effector size distribution. So high sigma means that it's more likely for us to get a mutation that has high impact on the trait value. And since we want to simulate the phenotypic adaptation, at some time point of the evolutionary process, we will put an optimum phenotype shift on one of the species. And our simulation, we did our simulation under different combinations of these three parameters. And at the end of each simulation, we extracted 100 individual sequences from each species. And the core idea to detect a molecular evidence in the genome is to see whether if there's a change in evolutionary rates, which you can think them as the accumulated number of substitution over a given time period. And within species, we're looking at the polymorphic site. The polymorphic sites are the positions in the genome that not all individuals share the same nucleotides, like the two colored columns here. And people ask the question that among those polymorphic sites, how many of them will lead to amino acid change and how many won't? Or you can say non-synonymous mutation versus synonymous mutations. And so basically that is saying, okay, so we can see here's a mutation from A to C in human 2, and we want to know if ATG and CTG lead to the same amino acid or different amino acids. And people take the ratio of PN and PS, and they think when PN is larger than PS, that indicates a weak selection because more amino acid changing or protein changing mutations are tolerated by the selection. But when PNPS is less than 1, that means the selection doesn't want any change in the original sequence. They just want to keep the original state, and that points to a strong selection. And for between species comparison, we're looking at the fixed sites. Fixed sites are the positions in the genome that all the individuals in the same species share the same nucleotide, but they're different between species. And people ask the same question, like among these positions, how many of them are non-synonymous and how many of them are synonymous. They also calculate the value of DNDS ratio. And now let's combine things together with MK-test. The idea of MK-test is to ask if selection left a detectable signal by combining the information from polymorphic site and divergent site. And they use a contingency table to compare the number of synonymous mutations and non-synonymous mutations at both levels. And they assume that when there's no optimum shift under neutrality, these two ratios should be about the same. But when DNDS is larger than DNDS, that's an indicator of positive selection. What's happening here is that most non-synonymous mutations are beneficial. So they quickly spread in the species and become fixed. And that leads to our observation of more non-synonymous at fixed sites than polymorphic sites. Or in other words, the adaptive change are driven to fixation and left a signal of selection. But when on the other side, when DNDS is less than PMPS, that points to negative selection. In this case, most non-synonymous mutations are deleterious. So they pop up as rare variants at polymorphic sites, but it's really hard for them to make fixation. So they just hang around at polymorphic sites. Only few of them can make fixation. And here's the results from our simulated data. Let's start with the baseline, the neutrality, no optimum shift. So all the optimum value are just a 0 for both human and chimpanzees. And here each line corresponds to different selection strands. So at the baseline scenario, we just play with the value of VS and sigma. And so the darker line indicating stronger selection. And on the x-axis, we have log scaled sigma. From left to right, it means the increasing of effector size variation. And on the y-axis, we show the value of PMPS ratio. So what we can see here is an clear pattern that as selection gets strong, as the effector size variance gets high, we see a lower trend in PMPS ratio value. That makes intuitive sense because the strong selection wants to keep the original state. They don't want the optimum phenotype shift to another value, so they just try to avoid any mutations that could change the original amino acid. And for the increasing value of sigma, because we don't want any non-synonymous mutations that would have high impact on the original phenotypic value. So we also try to avoid that kind of mutations. And for the DNDS value, we see a similar pattern here. That also makes sense because the same evolutionary force that works on those polymorphic sites to reduce the non-synonymous mutations also work fixed sites to reduce non-synonymous mutations. And now let's put the optimum shift. So the idea here is that at the time point of speciation, we let the optimum phenotype in humans shift to another value. So if the original distribution of phenotype in human population, if it is this peak, then when environment changes, the original population not adapt ideally, and the natural mutation will guide them to move towards to the new target. And in contrast, there's no change in chimp lineages. So the first thing we check is the number of substitutions that happens only in human lineage. So only a single lineage here. And so here's the number of substitutions happen across the generations. And our simulation starts with 10K generations of burn-in, and we put an optimum shift at generation 200K. So when there's no optimum shift, the number of substitutions just increase linearly as time goes by, which makes sense. And then we put a small shift. We let delta equals to 0.1, which is about the 0.7 times of the standard deviation. We didn't observe any change. Then we increase delta to 0.5. Now it reaches 3.5 times of the standard deviation of phenotype at equilibrium. Still no change. And until the optimum shift magnitude reaches 1, which is 7 times of the standard deviation of the phenotype at equilibrium. it is a huge shift. So basically, like to reach this shift, you have to kill everyone in the original population. And yeah, and we do see a burst of substitution at the time point of shift. And then it just returned back to the normal level after a short time. And of course, the higher shift magnitude will lead to a stronger burst of substitutions, but then they still go back to the regular level. And well, so of course, we can see the change in substitution rate at this time point, but most of the time in real life, we'll only have sequences from contemporary data, right? So in our simulation, that would be the sequences that we can get at generation 300K. And we apply MK-test on both sequences from human and chimpanzee, and we use the value, which is alpha, and it is the differences between 1 and the ratio of DNDS to PMPS. So when alpha is 0, that means DNDS is almost the same as PMPS. And so here each line, here we show the results from very strong selection, and we hope to see some signal. And each line here corresponds to different sigma square. And on the x-axis, we show the different magnitude, the scale, the shift magnitude. We scale it by dividing the standard deviation of phenotype at equilibrium under different sigma. So because the standard deviation of phenotype is different under each sigma, so that's why each line covers different coverage, different range on x-axis, but the pattern we can see here is whatever how large the sigma square is and how high the shift magnitude is, the value of alpha just sticks around 0. And we try to explain this. And in 2022, Hayward and Sella published a paper, and they investigate the allele dynamic under a rapid optimum shift. And they broke this process into 2 phases, depends on how quickly the phenotype responses to the shift. And in the rapid phase, the frequency of aligned alleles, those are the alleles that has whose effect size will push the phenotype towards a new optimum value. So they call them aligned alleles, and the frequency of aligned alleles just slightly higher than opposing alleles. And when it comes to the equilibration phase, the difference increases, but only subtle. And the key point here is that if you see, it is a cartoon, and if you see the number of green lines and the blue-purple lines, the probability for aligned alleles to be fixed is only slightly higher than the probability of opposing alleles to be fixed. So most of alleles, they just hang around at intermediate level or just get lost. And that kind of helped us to explain that why we see the value of alpha just sticks around 0 under different shift magnitude. And it is an ongoing project. So in our next step, we're planning to apply MK-test at different time points because we see a burst of substitution shortly after the optimum shift. So we wonder if the power of MK-test could be strong if we apply it at different time points. If we can see the power become stronger here, that would provide us some insights on what would be the best time for us to detect the molecular signal. Okay, to wrap up, so we try to build a bridge between population genetics and phylogenetics, or macroevolution and microevolution, to see if we can detect any molecular signals through PopGen stimulation. And at equilibrium, we do see the stabilizing selection's performance is consistent with negative selection, which kind of validate our stimulation. But when we put the optimum shift, we see we did see the signal, but the signal just fades away as time goes by. And so the current situation is that we don't see any evidence of molecular evolution under a large phenotypic adaptation between species under stabilizing selection. And I want to say thank you for all my lab members, especially Joshua Schreiber, who is the person who wears the pink shirt here. The idea is from him, and questions are welcomed.
So would you expect to have some signal if you had, continuous change in selection? Obviously, that wasn't just one shift, right?
You mean
Make your
multiple
path.
shift?
Yeah, I mean, there's, the conditions are continuously changing, right? There's not that the selection has stayed constant.
Yes, actually, we applied our simulation model on a real mammal trees with 61 species. We just place 3 points nodes which has a optimum shift. But the weird thing is that if we put the shift at a higher level on the tree, we can't detect any shift, like any diversity frequencies at the tip. That make us wonder, okay, if we just investigate the 2 species scenario, what will we see? So yeah, that's still under investigation.